Friends, today in this video we'll discuss about exogenous antigen processing and what are the important cells that plays an important part in this exogenous antigen processing and how this process takes place inside the cell. So at first look at this cell. These cells represent an antigen presenting cell where you can see that a bacterial pathogen that gets internalized and processed and presented with MHC molecule on the cell surface. Okay. So the basic thing that occurs in this antigen presenting cell is that at first antigen gets recognized. So how this antigen gets recognized? Which signal dictate the cell to internalize this pathogen? And there are several receptors that are present in the cell surface, and upon binding of this bacterial pathogen to the cell surface receptor, it starts its internalization process. So those receptors that are present such as CD40L, this chemokines receptor, all these receptors that are present on the surface of this antigen presenting cell, when they get binded by this bacterial pathogen, they start internalizing this pathogen. And once this pathogen gets internalized, it gets processed via the lysosomal enzyme or the cathepsins that are present in the late endosome that will cleave this pathogen into small parts, and finally they will be presented on the cell surface upon combination with this MHC molecules. Okay, so these are the MHC molecules, these green ones. So once they get binded to these MHC molecules, they are presented on the cell surface. So those cells that participate in such events are the antigen presenting cells and macrophage, dendritic cell and B cells, which I have written here. These cells are the antigen presenting cells that are present in our body. So these are the three basic steps: the recognition of antigen, okay, the processing and the presentation. So these three step rather plays a very important role in this exogenous antigen processing. So once these processes are done, finally, so mainly the T helper cells comes and recognize this MHC2 molecules that are associated with these peptides, and finally they will start doing the process of this clearance of this bacterial pathogen from the body. So this is the basic mechanism of how exogenous antigen processing is done. So in the next figure what we will discuss is that how this process takes place. So as I have said in the previous slide, that these processed peptides are presented together with MHC2 molecules on the cell surface. So how does this MHC, or where from this MHC comes from? MHC molecules are synthesized in the ER, and upon synthesis they get assembled inside the ER and stabilized via this invariant chain, which is a trimeric protein that binds to these MHC2 molecules and stabilize them. MHC2 molecule has a very higher recognition of binding protein, so it is very important that after its synthesis and its assembly, its binding site must get binded by this invariant chain. If it binds to this MHC2 molecule binding cleft, then no self antigen will bind here. Okay, so MHC2 molecule binding cleft has a very higher tendency to bind the peptides. So this this invariant chain binds to this peptide cleft and stabilizes them. So once these MHC2 molecules are assembled, once they get stabilized, they start moving from endoplasmic reticulum to Golgi body, and from Golgi they moves to late endosome, and then from late endosome they moves towards cell surface, which is the basics of secretory pathway. So after it moves from ER to Golgi and then from Golgi to this late endosome, because late endosome has acidic pH, and in this acidic pH there are certain enzymes that gets activated and they start cleaving this, such as cathepsin. What they do, they start cleaving this invariant chain, leaving a small portion of this invariant chain to remain binded to this MHC peptide binding cleft. So this cathepsin, you can see here, they have degraded the entire chain and they have left only this small portion of this invariant chain, that is what we are calling as clip. So again, this presence of clip is very important, as if you mutate the gene that encode for this clip protein, in that case what will happen, before the binding of any foreign antigen, this MHC2, which has a higher tendency of binding your protein molecules, they will start binding the self antigen, and once it will start binding the self antigen, then autoimmunity disorder can result. So this clip plays a very important role, and if you mutate the gene that encodes this clip protein, in that case what will happen, this MHC2 molecule will start binding the self antigen. So it is also very necessary for this clip protein to function. Okay, so after this clip processing of this clip protein take place, the foreign bacteria that has been internalized via this phagocytosis, they comes into this early endosome, and from early endosome they move to the late endosome. Okay, inside the late endosome, this foreign protein or the pathogens are cleaved via this cathepsin, and this cathepsin cleave them into small part. The protein gets cleaved into small part, and once they are ready, once they are cleaved and ready to bind this MHC2 molecules, this DM protein, they remove this clip from MHC2 molecule and allow the binding of this foreign protein to this MHC2 binding cleft. And once they bind here, they are ready to get presented on the cell surface. Okay, so any protein that is coming from outside, internalized via phagocytosis, they move from early endosome to late endosome. Inside the late endosome, cathepsin start degrading this foreign protein, and this foreign protein will bind to this MHC2 molecule only when this clip is removed. This clip is removed by this DM protein. DM protein remove the clip, allowing the binding of this protein foreign part, and then it will move toward the cell surface, where it will present this peptide to these T cells, and then the entire process will start to remove this foreign particle from the body. So let us just summarize this. So what happens in the endoplasmic reticulum, the MHC2 protein gets stabilized and also get assembled. They get stabilized via this invariant chain. Then they move from ER to Golgi, and from Golgi to late endosome. Inside the late endosome, because of the acidic pH, the invariant chain gets cleaved, leaving a very small part clip, and this clip covers the peptide binding cleft of MHC2 molecule until the foreign peptide will come and bind there. So the foreign peptides come from the outside. They are phagocytosed, and once they comes into this late endosome, they get cleaved by this cathepsin to smaller peptide fragment. Once these smaller peptide fragments are ready to bind to this MHC2 binding cleft, the DM protein removes this clip, allowing the binding of this smaller peptide fragment to this MHC2 peptide binding cleft, and once they bind here, they are ready to get presented on the cell surface, where T cells will come and recognize them. So this is how the entire process takes place of exogenous antigen presentation, and I hope you guys have understood the entire thing. If you did not understand anything, you can put your question in the comment section, and thank you for watching this video. In the next few lectures we will discuss about how these T cells and B cells are activated and how do they function. So thanks for watching this video.